Okay, so we are ready to start with the second talk. Patrizia Fattori will present vision at the service of action control in the medial posterior parietal cortex. So I leave the floor to you.
So, hello to everybody. I'm really happy to be for the first time in my life to this very rich meeting, scientifically very, very rich. And I confess that it is years that I'm thinking to come to this meeting. But always I refrain because this meeting has the only deficit of so close to VSS. So next week there will be a huge international meeting and the close coincidence lowers down the ability to organize everything. So this time, I thank the organizer giving me this opportunity, and also after speaking to poster presenters, I realized that it is so rich that I will encourage. Why is it going back and forth? Okay, we use the cone. No problem with the cone. No, faccio col cuore, non c'è problema, così posto. Ok, dopo quello lì serve. Sì, dopo ci uso questo qua. Ok. For the questions, I will leave the cone to you, but now I will use for a while. So, let's start with the topic of my presentation, which is focused on the posterior parietal cortex. Mainly you will see in the medial part of it. So I will start by separating the inferior and superior parietal cortex in the macaque, and then I will give you a lot of information about an area of the superior parietal cortex, which is area 𝗍𝘁𝗁, in which I will show you some aspects of analysis of visual information, but always at the service of action control. And I will finish by briefly showing you some aspects of area 𝗍𝘁𝗁 in the human brain, but I have many, many slides, so for the questions I'm open to increase any aspect of my presentation. So let's start with a very short history of the posterior parietal cortex, which, you know, belongs to the dorsal visual stream as reported now in every textbook of neuroscience. The story of posterior parietal cortex started about 50 years ago by going into deeper details about the organization of this huge part of the cortex, which is interposed in between the somatosensory cortex and the visual cortex. This is a map done by Rizzolatti and colleagues showing only the areas exposed on the surface or in the interhemispheric face of the parietal lobe, but we have many more areas into the sulcus, as I will show you in a while. The study were first addressed by Vernon Mountcastle, which was a wonderful neurophysiologist that did several penetrations, microelectrode penetrations in the primary somatosensory cortex, and then he went a bit caudally in Brodmann area 5, and he was the first person showing in the 70s that we have very nice joint-related discharges. In any repetition of the passive movement of the joint of the monkey, he had a very repetitive response. And this was the first demonstration of a kind of somatosensory representation which went farther back with respect to the primary somatosensory cortex. Then, okay, this is just to quantify that indeed he found that more than 99% of the neurons had some somatosensory related properties, and for the visual properties, you see you have less than 1% of the neurons, so very, if nothing visual in the superior parietal lobule from Vernon Mountcastle study. Conversely, his penetrations in the more lateral in Brodmann area 7, which corresponds to this part of the brain, he found many visual responses, so neurons like this one, in which when the monkey was keeping fixation, this is the trace of eye movements, repetition of visual stimulations evoked a very nice repetitive response. And the similar table, but the opposite with respect to superior parietal lobe, because you see that joint neurons were about 1 or 2%, and all the other neurons were visual. So after these first studies, it seems that inferior parietal lobe is the place for analysis of some kinds of visual information, and superior parietal one for somatosensory information. The study went along in the years, and this is a map derived from Ivarian laboratory, a bit more later, 82, in which he found, you see here, visual black and visual and somatic white neurons in the inferior parietal cortex. So again, the inferior parietal lobe is a place where you find visual information and also somatosensory one, but nothing was said so far for the most medial and posterior aspects of the posterior parietal cortex. We needed to arrive to the late 80s, and 𝗙𝗁𝗵 was the first one in addressing the studies in this part of the brain. So if we cut a parasagittal slice of the monkey brain and we see it in this way, so this is the occipital lobe, this is the parietal lobe, this is the parieto-occipital sulcus, very, very medially, so at this side, you find that she had penetrations in this deep part of posterior medial parietal cortex, and she found very nice visual receptive fields. These are represented in a way that this is the fovea, this is the horizontal meridian, this is the vertical meridian. She found a representation of the inferior contralateral visual hemifield in this part of the cortex. With Professor Galletti in Bologna, we started the study of this part of the brain in the 90s in awake monkeys. Before, I forgot to say that 𝗙𝗁𝗵 was studying anesthetized monkeys, which at the time were the most common experimental paradigm. And what we did is we had penetration starting in the occipital lobe through these areas that are named here, and then jumping down into the parietal lobe, similarly to 𝗙𝗁𝗵 approach. And what we found is that in the primary visual cortex, V1, we had very small receptive fields close to the vertical meridian, inferior vertical meridian, this is the fovea, as typically known for this part of the primary visual cortex. Then we had the electrode going down into area V2, and we have a slight increase of size of receptive fields, and deeper, further deeper, once we jumped into the parietal cortex, because this is the parieto-occipital sulcus, so now we are in the deep part of the parietal cortex, we found very huge receptive fields, much bigger with respect to what we expected from an increase in size going to higher eccentricities. And we call this area V6 because at that time it was already known that there was V5 and V5A or MT, MST, and then with Samir Zeki, this was the time to call it V6. It was the sixth visual area known so far. And going further down with the electrode into this area, you see that receptive fields move quite orderly from the inferior contralateral quadrant to the horizontal meridian, and then going further down, very deep into the medial surface of this parietal lobe, we have receptive fields going up in the superior contralateral quadrant till the upper vertical meridian. So it is a very ordered spatial representation, retinotopic representation in this part of the cortex. So area V6 is a clear retinotopic extrastriate visual area. And the electrode was not so deep into the parietal cortex, but a bit higher up. So it reached the anterior bank of parieto-occipital sulcus, but up, not so down. We had a different situation. We have very huge receptive fields as before, but the order of the representation is not retinotopical, of the retinotopical type, because we have, you see, a first receptive field on the inferior vertical meridian, then a second one on the horizontal meridian, but the ipsilateral visual field, and then two more receptive fields in the inferior contralateral quadrant, jumping from one position to the other. There is no more order, no more retinotopic order. in this part of the cortex. And additionally, the visual stimulations that we needed to use to activate visual responses were much more complex than for area V6 or other visual areas of the time. So for V6, it was enough to have spots, either dark or light, dependent from the neuron, or bars moved on the receptive field, or single borders, either dark or light borders, always moved, the arrow indicates movement on the receptive field. For area V6A, some of the neurons responded to these stimuli, but not so many. Some of them needed more complex visual stimulations, like corners moved on the receptive field, so different angles present on the receptive field, or parallel lines like those evoked by shadow of the hand, the fingers moving on the receptive field, or bars enlarging or decreasing their size. So we have a more complex visual representation in this area. And additionally, even using these complex simulations, we were not able to activate about 40% of the cells. We don't say that they are not visual, but they were not activated by our visual stimulation. So it seems that this area is a bit more complex in the visual analysis, let's say, or representation of visual information. So with this data, we proposed a new frame for the visual properties of superior parietal lobule that since before Colby, it was considered to be somatosensory, while the inferior parietal one was both visual and somatosensory, as I summed up very, very quickly. But after these studies, and now we have a view with a partial opening of parieto-occipital and intraparietal sulcus, to show that we have many more areas, not only V6A. I don't have the time, but if you have questions, I have material also on these aspects, in which we had both visual and also somatosensory representation. So a little update on the knowledge that probably you have already seen, but just to make a common frame for the data I will present. So I want to stress that in the first representation of the dorsal visual stream proposed by Ungerleider and Miskin, the arrows were pointing to the inferior parietal lobule, because at that time this was the frame I was showing. And they spoke about a wire system, and Goodale and Milner updated this view with the concept of how, so not only spatial properties analyzed there, but all the attributes that are needed to organize actions, not only the spatial location, which is of course an important attribute. And what it became clear about 20 years ago is that we can split the dorsal visual stream within, at least at that time, two streams, one more medial that brings visual information from the occipital lobe to the superior parietal one. And we like to call it a dorsomedial visual stream, just to differentiate from the other more lateral aspect of the dorsal visual stream. So what about the motor properties of these parts of the brain? Again, the first one, the pioneer in this was Moncassel, that trained the monkey to perform reaching movements toward targets that were located in different positions in front of the monkey, and he found these very nice and repetitive discharges connected, temporally related to the action of the reaching movement of the animal. After Moncassel, many have used delayed reaching tasks in the superior parietal lobule. For example, John Kalaska, Roberto Caminiti, Johnson, many others. If I forget some names, forgive me, but there are many laboratories that have studied spatial related properties for reaching in superior parietal cortex. I showed just one example neuro from our lab, in which the monkey was trained to reach one out of three targets at the same distance from its body, from its gaze, and the activity is aligned with the onset of the reaching movement. And you see that in this area, very medial in the posterior part of parietal cortex, we have very nice peaks of activations related to the occurrence of the reaching. And these peaks are tuned according to the direction of the movement. For this particular neuron, the best direction was toward the left target. Then we went on by producing a paradigm in which we also checked for our movement amplitude, which was not granted because usually the most typical task in which many researchers, also in the frontal lobe, for example, all of you know 𝗙𝘁𝗁𝗶𝗶's work in primary motor cortex when he found this spatial tuning of reach-related discharges. He was the first one, but after him, many researchers used a center-out task. So the monkey was starting from a position and were reaching different directions from a central one, but always on from the same distance to the body. What we did is that we also used this apparatus in which the targets were also located at different depth, different distances, so we could also check for our movement amplitude. And I show you here just 3 of these responses to this central line, so to the near pink, central yellow, and far red targets. And you see that the peak of reach-related discharges is also tuned to the amplitude of the movement, which is another parameter which is encoded here. And of course, not only here, but it has been tested in this part of the brain. What we did more recently is not only to correlate, like done so far, the discharge with some events. To come to Alfonso's question to Luca before, he was saying, he was asking whether what is a response of a neuron. In this case, we can correlate the discharge of the cell with the occurrence of a certain act. What we did more recently is to try to decode from the neuronal activity some parameters of the task. Is the different epochs of the task, not only the movement one, but only the delay before the movement or a free epoch at the beginning when the monkey was free to look wherever he wanted and also was immobile with a hand on a button near the belly. But also the holding time when the monkey was holding the target for a period. And you see that from the activity of 𝗍𝘁𝗁 we had a very good decoding performance. And for the epochs, but also for the spatial locations of the targets. These are the 9 targets that are encoded. This was the home button, so the initial position of the movement. And you see that with a very good accuracy, about 80%, we could decode these spatial locations for reaching from neuronal activity. If I could say, we had, as Luca, we did not check each other, but we are on the same page when we speak about mixed selectivity. In this case, in 𝗍𝘁𝗁, so different parameters of different tasks that activate the same neuron. So with these different colors, we can indicate different parameters, for example, the location of the target, the epoch of the trial, or the direction of gaze, just to mention 3 of them. And you could see that you have a different weight of the different parameters, but neurons that are responsive to more than one of them. So it is a rich place where to study the different computations that are going on when we perform our everyday activities. So this is the picture of the 2013 version of the Candle book. I just enlarged the words, but these are those originally presented in this book, in which it is clear that the areas of the dorsomedial visual stream, including 𝗍𝘁𝗁, strictly connected with dorsal premotor cortex, is a circuit for reaching. This was suggested at that time, while the connections between the areas of the dorsolateral visual stream, so the dorsal lateral parts of parietal cortex with the inferior premotor cortex, is the circuit for grasping. And I would like that you follow me in seeing that we have not only reaching in the medial parietal cortex, and we dared to test this more than 10 years ago by proposing to the monkey 5 different objects in a stable location right straight ahead from the monkey, So they were proposed one at a time, and the objects evoked different grip types. And the monkey had to perform a reach to grasp task. So he had to leave a position close to the body to reach the position, constant position in the peripersonal space, but changing the grip time to perform. And we found example neurons like this one, in which we have a peak of activation, this is movement onset, when the reach to grasp movement occurs, but this activation changes according to the different grips performed, with, in this case, a preference for the more precise and complex grip, which is to catch this little cylinder into the fixture. So you need to oppose the first phalanges of the first two fingers. If you don't do this, you are not able to grasp it. But another neuron could have exactly an opposite discharge. In this case, this cell does not respond to any of these two grips, but is maximally activated by the more rudimentary grip, which is a whole hand prehension. You can use any finger you like with any orientation of the wrist in order to grasp this ball. And this was the preferred grasp from this cell. Of course, as Luca was pointing, one neuron is not enough to make a story, so we have analyzed the entire population that we could study. And we found that about 60%, 70% depends on the task phase we analyze, of the neurons. So the great majority of neurons in V6A are tuned for the grip types. So this is the population discharge aligned on the onset of the movement, and you see that from the best to the worst grip for each individual neuron, we have a huge discrimination in the population. So the discharges can very well differentiate among the different grips. So this was at the time the first demonstration that not only reaching or special parameters for reaching are encoded there, but also parameters of object shape, I will come to this later, in order to organize different grips. And also, as we did for the reaching, also for the grasping, we have good performance in decoding with different systems. In this case, we have used an explainable system based on convolutional neural networks. Don't ask me more details on this. And we found that these in two different monkeys, we have very high percentage of accuracy in decoding the different grips that the monkey is performing in our task. So with this, I think we can update the 2013 view of Candel by cancelling the word reaching for this medial circuit and speaking more about the reach to grasp circuit. And I make you happy showing that the most recent version of Candel shows this picture. Without naming anything, it just says macaque monkey, areas in the cortical convexity, areas inside the parietal sulcus, and it places many arrows without mentioning any role. So it's more open to our other interpretations. So far, I have shown you recordings in tasks like this one, in which the action occurred always in the dark. Even before, I did not mention, when the monkey was performing the reaching, he had just a very small light visible for the entire duration of the task. It was not on a screen, it was a real object, so there were no visual cues giving some special information like the border of a screen, for example. Because we wanted to be sure that the modulations we were seeing, like, for example, these peaks of activations, were not due to visual confounds, were not due to the fact that the moving arm was entering into the visual field, because I've shown you before that any moving object, not any, some objects moving on the receptive fields could activate visually the cells. So we wanted to be sure these were not visual responses. So we had the tasks, either the reaching or the grasping task, completely in the dark. For the grasping task, the monkey had the possibility to see the object before, at the beginning of the trial, and then lights were off, so he had to perform the grasp without any visual feedback. We decided to go deeper into these aspects of the interaction between visual information and movement-related signals. And so we proposed to the animal to perform reaching movements either in the dark like before or in complete light, so we could see the environment and also the moving arm. And I show you neuronal discharges in this way. With the gray, it is in the dark. With the white, it is in the light. So this is one neuron, and these are 3 other different examples. The first one, you see as a discharge only in the light. This is the onset of the movement, and we have practically nothing occurring in the dark. So we can assume that this neuron receives only visual information related to the moving arm, and does not receive anything related to the somatosensory information or to the movement-related information of the arm that is moving. The second neuron is exactly the opposite. You see, it has the same peak of activation, or not significantly different, when the action is performed in the dark or in the light. It means that vision does not add anything to this cell. This is driven only by motor-related signals. For motor-related, we can mean also an efference copy of the moving signal given by frontal cortex to the limbs. And these 2 other neurons, indeed, have both kinds of information reaching them. The first one has a higher peak of activation when the movement occurs in the light. So you can assume that it receives a corollary discharge, but also a somatosensory input, and additionally visual input, signaling what is going on. And this is quite easy to be understood. The other neuron too receives these 3 kinds of inputs, but in a subtractive way. So we can assume that the motor-related discharges, being it corollary discharge or whatever, are exactly the same in the 2 conditions. But vision will diminish the activation. It is subtracted from the activation of the action performed in the dark. And we found exactly a similar attitude, a similar population discharges when we perform the grasping actions either in the dark like before or in full light, similar to reaching. And these are 4 neurons. One reached only by visual inputs related to the moving arm. These by both only by motor inputs because it does not differentiate. And these 2 are summing or subtracting visual input from the motor-related one. So we reasoned that this kind of visual motor neurons could be very useful to play a role in the movement control basing on feedback on or feed-forward inputs. So by counting differently on the expected outcome of the movement given by these somatosensory, somatomotor discharges, and the visual input that is coming from what I'm doing, maybe they can have a role in organizing our action, in controlling if the actions that are organized by the frontal lobe are going on in the right way. So at that time, thanks to this complex interplay, we proposed that 𝗍𝘁𝗁 could act together with many more areas in the brain, to act as a state estimator so that it could compare information coming from the visual cortex or somatosensory cortex that give us an information about the expected outcome of our action with a signal delivered, for example, from dorsal premotor cortex, and in a while I will tell you why I'm thinking to dorsal premotor cortex. So signals from frontal lobe about movement planning that could allow this neuron to predict the consequences of the action. And of course, 𝗍𝘁𝗁 can be in the position to inform dorsal premotor cortex if the action is occurring correctly or not. Of course, this is a speculation. We don't have the proofs of that, but it is something that we derived from the functional properties I've shown, but also from the connections that by injecting neuronal tracers in area 𝗍𝘁𝗁, but also in other areas of the brain, for example, those in the frontal cortex, we found direct links. So it means when you see one of these lines, you see that there was a significant number of neurons, more than 20% if the line is thick, or less if the line is thinner, that had a direct anatomical connection with one of the areas that are mentioned. And you see that we have some areas of the occipital cortex of visual areas, not primary visual cortex, but V2, V3, V6. Also areas like MST, V4, areas with visual properties in the lateral parietal cortex, but also areas with visual properties in the intraparietal sulcus, but these areas have also oculomotor properties, reach-related properties, somatosensory properties. You see that area AP that Luca has shown you before has some direct connections with V6A, not so strong, but are present. And also, of course, the areas here in the frontal cortex, particularly strong is the connection with area F2 in the dorsal premotor cortex, but also F7 of the dorsal premotor cortex. So you see that this cortical circuitry puts V6A in a loop in which most of these speculations can find an anatomical counterpart. But of course, this, as I told you, is open to discussion. So let me now go to the last part of my presentation, in which I would like to show you something more tied to the object representation. So we showed 5 objects in a context in which we had a barrier. Our barrier was not visible as the one of Luca. It was close to the body of the monkey. He knew that it was there, he could touch it, but he did not see it. It was not in the visual field because we came from the visual cortex. We wanted to be sure that there were no differences in the visual reflection of anything. So the objects were exactly seen in the same way, but the monkey knew he could not reach them. He has just to push and pull the button. If he was moving, it was not rewarded, and he had just to keep the fixation on this little light on the top of the object. So also eye movements were fixed, and you can check the gaze down there. So, and we found very nice object-related discharges with particular preference for individual neurons. We checked in the entire population, which is the feature encoded by these neurons. Even if we have only 5 different objects, we have used a hierarchical cluster analysis in which objects that evoke similar responses are placed close together. And we found a first cluster with the ring and the ball, another close cluster formed by the plate and the handle. And then we had farther away the stick into the groove. So we found, although we have just 5 objects, a kind of visual code in this representation. So we have here the round object, and then the flat object, and the very complex object alone. Then, going in this direction, we studied what was going on if at the beginning of the trial we show an object, for example a ball in this case, and then we have complete dark, we perform the action in the dark, and we perform the grasping here. And this is the discharge of one cell to 5 different object presentation and 5 different grips. And you see that in all cases you have a double peak of activation, one for object presentation and one for grip occurrence. And we again perform the hierarchical cluster analysis. So this is the object illumination before the grasping. And we found a cluster of the ring and the handle and another cluster of the other 3 objects. And then we perform the same analysis on the same cells, but in the movement-related epoch, and we found a different clustering. You see, we have a cluster of the ring, plate, and stick in groove, and then the handle and the ball, which is farther away. We found in this a motor code. Because we have here, for these 3 objects, you need to use the index finger in order to perform the grasp. You cannot do it correctly without using the index finger. But you could grasp the handle by inserting any finger inside the hole, and the ball you could do whatever you like. You don't need to control very finely the index finger. So we found in this case a motor rule. So we saw that in the same trial, the same neurons dynamically changed their encoding in the population, of course, not only individually. And we saw a visual encoding, a visual motor encoding, if you like to say, at the beginning, and then a motor encoding later on the trial. We also looked in the delay epoch, so when there was no more object visible, but the monkey knew which kind of action to prepare, and in the delay epoch we found again a motor code. So it seems that we have The population that at the beginning of the trial has a kind of encoding of objects that as soon as the object is no more visible and the monkey is preparing the action that he will perform later on, there is a switch to a motor code. And going on in this direction, we also had a study in which the monkey that was trained to grasp the objects was just shown two of the most similar objects, the handle and the plate. So seeing laterally, this is the real photo of the original objects. They are very different, but from the monkey's point of view, they are very, very similar. You see that you could recognize that the plate is a little thinner in this part with respect to the end, but they are visually very, very, very similar. And what we found is that we have neurons like this one, in which you have a strong discharge, in this case for the handle, and no discharge at all for the plate, despite the similar appearance of the object. We suggested that this was a different affordance that was encoded by the neuron. And we had a demonstration of this by proposing to the monkey to grasp the usual thinner version of the two objects and a thicker one. So we had a thick handle and a thick plate. Of course, the handle had a hole, the plate did not have the hole, and we found that the coding did not change. So you see that the handle, either thin or thick, evoked the same discharge, and the plate too. Thin or thick did not matter, and despite the different appearance, the discharge was the same. So we suggested that this kind of discharges could be useful to encode the shape of the objects for action, not the visual attributes per se. The last data I would like to show you is about the mirror neurons in 𝗍𝘁𝗁. You knew everything from Luca before, and we proposed the monkey to perform his task. We have here a cut in the middle of each discharge to show you the discharge at the beginning when there was the presentation of the object, and here aligned with the onset of the movement. And you see that, of course, we have a nice tuning of this discharge according to the different objects and grips. And then we had the experimenter performing the same action, and you have this same neuron discharging in a way to resemble mirror neurons, but differently from the original definition of the mirror neuron. You see that you don't have congruency between the discharges in the motor situation and in when the object appears or the action appears in this context in which the monkey is acting, on when the experimenter is acting. For example, we have here a very nice peak of activation when the monkey grasps the plate, but no activation when the experimenter does it. Okay, we still have a congruence for this grasp. The little ring is grasped in the same way. No, is evoking the discharge in the same way when it is grasped from the monkey or from the experimenter. But we don't have a perfect matching between the discharge when the action is observed and is executed, as Luca was pointing. And here we had a third condition in which the object was presented without any possibility to act, again an unseen barrier. And we have an encoding which is somehow different with respect to the one found. But first, let me tell you that the mirror neuron population as this discharge, collectively speaking. I must say that they are a minority of neurons in 𝗍𝘁𝗁. They were about 20% of the neurons. So while the majority is grasp-related, the mirror neurons are very few. And you see the peak of activation when the action is performed by the monkey and the peak when the action is performed by the experimenter. We must say that the peak is delayed in this case because the experimenter was much less smarter than the monkey. The monkey was quicker. The experimenter was a bit longer in performing the action, so the movement was longer. But I leave the mirror neuron stuff to let you concentrate on the discharge that we have when the object was presented in the action execution condition, action observation condition, and no action condition. And we compared in the population the discharge in these three conditions. And you see that the objects evoked different discharges only when observed, but in the three different contexts, with the highest discharge. Of course, they were normalized discharge when the monkey is later on performing the grasping on this object. And the lowest discharge is when the monkey knows that is the experiment that is going to grasp it. And also, these confusion matrices you see in the three conditions are organized in different ways, so indicating that the dynamics in the population for object encoding is different in the different contexts in which the action occurs or does not occur. The context in which the monkey knows the action is going to be performed or not performed. So I finish by showing you that we have found the location of the human 𝗍𝘁𝗍, and this is for comparison the macaque one. This is the one in the human, maybe it's better this medial view, in which you see it is located in the top and posterior part of the medial side of the parietal cortex, and we have activated it with many stimulations, visual stimuli, reaching and grasping, and we were not the only one studying this area. With different names, it has been studied by many other groups. So just to compare, this is also Mel Goodale involved together with 𝗍𝘁𝗁𝗁 in finding an activation in this study for reach-related discharges in this area that was called anterior SPOC, located on the anterior bank of the parietal occipital sulcus. And this is another study by Gallivan in which the SPOC was activated for performing grasping actions. So again, we have a parallel organization of this part of the brain in the macaque and in the human. So I conclude by summarizing that we have many interesting things going on in the parietal cortex, also in the medial parietal cortex, not only in the lateral one. And we have analysis of visual information for many different tasks that we can perform in everyday conditions, like reaching or grasping, either in peripheral or in central vision. I did not have the time to present this data, but also in more dynamic context in which we have, for example, to select and grasp an object in a supermarket shelf. And I finish by showing all our grants and our group that has worked for decades on this topic. As you can imagine, there is a huge effort behind this study, so I want to give the credit to the research group. So thank you for your attention. I finished.
Okay, time for questions.
Maybe you need this one. I will take the other one. I give to you.
Yep, okay.
Hi, thanks a lot. That was very interesting. So I'm interested particularly in this neuron that seems to change their selectivity in different moments of the task, when it's just seeing the object compared to grasping. There was this tree structure that you show, like a different clustering.
This one?
Yeah. So what is the computational explanation for neurons that behave like this? So why one neuron should encode something in a way, I don't know, 1 second before and then change completely 1 second after?
Yeah, in this case, we can just speculate. We don't have proofs of that. That at the beginning of the task, so in this case, when the monkey is engaged in capturing the information of the visual object, maybe useful for the grasping or for other things, the neuron is starting some kind of computation that change. In the moment in which he has, for example, with the light off, he has to remember the characteristics of the object, for example. It's a working memory that keeps trace of the shape, because in 2 seconds' time, 3 seconds' time, he has to perform a particular grasp and not another one. Other one, it will be too late and he will not receive the reward. So maybe it can be a switch in the encoding that is useful at the moment to perform the action, and probably a second later is gone. We did not check this, of course, but this is just a speculation. And I also may add something in this regard. What is peculiar is that the same neurons if we compare the analysis done with just observing the object in the barrier task, no grasps allowed, and the epoch I'm just commenting now at the beginning of reach to grasp task, so it's only vision in both cases, but this is outside the grasping context. This is inside a future, immediate future grasping. The encoding is similar, but not exactly the same. It seems that probably the neuron is in a lazy way, okay, it's a plate, I don't care, I just wait for the signal to release my button. And now it's, oh, it's a plate. I have to keep in mind that I have to grasp in this way. But it's just...
Thanks. So, did someone try to do some optogenetics or something to knock out this neuron and see whether they make a difference in different stages of the task?
It would be very, very interesting, yeah.
No, nobody did it.
No,
right? We don't have causal data on that. Okay.
There are so far very few studies with optogenetics in the awake behaving monkeys, and very brave, difficult studies. We did not dare to do it.
Thank you.
Thank you for your questions.
Congratulations, it was really very beautiful talk. It's a bit of a sort of parallel topic that I'm raising. Did you have any chance to record from the precuneus?
No, I did not.
I don't know if you know why I'm asking this.
Yeah,
because we had relatively recently found that the patients who have amyotropia and can show some blind sight for discriminating the orientation of visual stripes, and for discriminating the movement. But they didn't have any conscience of that, right? So I wonder, and we found that by looking at the resonance of structural resonance of the patients, and those that had more spared, I'm not saying that it was intact, but much less damaged lesions, it was on the precuneus. And it is a reasonably, well, for clinical, it would be a small number, let's say we are 12 or 14, I don't remember, patients. But for us, coming from experimental things, it was reasonably. So I wonder whether this could be, how does it look to you?
No, it looks perfectly in line with what I think, because if we see the anatomic location of 𝗍6A, I think that in all the case of patients, also I have in mind patient MC that has been studied by Jody Callum, Simona Monaco and others, Arcaro is the first name. She was impaired in all the tasks related to the ventral visual stream because she had a massive occipital lesion. She had spared, at least on one side, this part of the brain, and she could work well in the tasks in which she need to use motion information or visual motion, and motor information to perform her tasks. So I think this is in line with what you are finding. I think this part of the brain has much more to show in the human than expected till recently. So.
Yeah, because this processing, of course, can occur. Can work even without conscious, no?
Yes,
so
yeah, exactly. This is what these patients are doing. They are grasping flying balls with very high accuracy and saying, oh, this is the little one, or this is the big one, because it feels optically that it is the small or the big. But she says, I don't see the ball and I don't grasp it, so it's perfectly in line.
Thank you.
Brett?
Well, thank you for your talk. So I have a question actually also about this notion of, well, you showed that context modulates the responses, and you know, as I understand it, the With the animals, the grasp itself is essentially the end. They grasp and they're done. But you know, in the wild, the grasp is just a means to an end to then enable a subsequent manipulation of the object. And so for the same physical object, you might grasp it in different ways according to what you're going to do with it after you grasp it, this notion of end state comfort. So I'm just curious if you think that processing in 𝗣𝘁𝗍 is downstream of computations having to do with end state comfort, or if 𝗣𝘁𝗍 is providing all of the possible visual motor, you know, biomechanically possible grasps to an object, and then subsequent processes reduce that set to what is actually aligned with the goal.
Okay, I can just speculate. Don't take notes. I think it's downstream. I think that the premotor cortices are sending 𝗣𝘁𝗍 information on what has been organized in order to check if it is going on correctly, not in order to choose what is the best choice. You are referring to the elegant studies that 𝗤𝗁𝗁𝗍 and his lab performed in the lateral part of the parietal cortex, in which the grasping was the first of following action. So they trained the monkey to grasp and to place the object somewhere or to bring back to the mouth. And they showed neurons able to, just for the grasping, to differentiate the final goal. I think that these computations are more done in the lateral grasping circuit, not, but just a speculation. If somebody has a different opinion, I'm happy to discuss.
Thank you very much for the clear and interesting talk. I have a question about something you said in the beginning and in the end, and I have difficulty reconciling it. In the beginning, you said that in 𝗣𝘁𝗍, as far as you showed, most or all the receptive fields are quite peripheral. And then in the later part of the talk, you say that there are a lot of object properties of the target being represented. If I have to do a complex grasping task, I would look at the target. And so it would make sense to also have foveal receptive fields in that area.
Yeah, you're right. The penetration I showed is with peripheral receptive field. Thank you for the question, otherwise I could be misunderstood. But if I show you here a heat map of all the receptive fields in 𝗣𝘁𝗍 superimposed, this is the fovea, and the more you see red, the more receptive fields we have. So these are more than 200 receptive fields located here perifoveally in the inferior contralateral quadrant, some of them more peripherally, like the penetrations I showed. And when you see, maybe, yeah, you can see these light colors, we have some receptive fields in the upper visual field and some of them in the ipsilateral quadrant, but the majority are here. So we have also a huge foveal, perifoveal representation. And also we try to superimpose this representation with the trajectory of the reaching movements that human participants were performing to foveal targets that were located everywhere on a frontal plane. And we found that the trajectories of these participants, if we superimposed the targets in a central position, but they were everywhere, you see that the trajectory of the arms were mostly coincident with the visual representation that we found in 𝗣𝘁𝗍. So we did this, of course, for the reaching, but I guess that when you, at the end of the reaching, you grasp, of course, you need the attention to be there and the gaze also to have this foveal representation in order to insert the finger into the little fissure and so on.
All right, thanks a lot.
Thanks for the question.
So there is a question here.
Oh, thank you so much for the talk. You've mentioned that, and many people have mentioned that this 𝗣𝘁𝗍 area is working on a reach to grasp. And is it really reach to grasp? Is it reach and grasp? So your data, I don't know, for instance, if you just show, if the object was in front of the monkey, really by its hand, no reach was involved, would you still see this to fire? So is it really reach to grasp or is it grasp?
Yeah, this is an excellent question. we never tried to perform grasp without a proximal, let's say, component of the movement. So we don't have a clear answer. What we guessed from comparing the properties of 𝗍𝘁𝗍 with those of AIP in the lateral grasping circuit, if I can find, because I have so many slides that going one by one, it will take me time. But we compared the connections of these two areas reported in the literature and from our own work, and we found that, I will find it, okay, I will escape from this mode and find the slide and come back to you, sorry for a while, okay. Okay, don't be scared, I told you that I have many, because I knew there was so much time for the, okay, now I found the right, okay. So in this case, I'm just comparing the amount of connections. So I don't have a straight answer to your question of 𝗍𝘁𝗍, which is pink, and AIP in the lateral part of the parietal cortex. You see that they are directly connected, not so much, but these are cells in AIP that send their axons to 𝗍𝘁𝗍. And these are reciprocal connections, so in other works we have seen the opposite. And comparing data with Lupino and Borra laboratories, you can see that 𝗍𝘁𝗍 has much more connections with extrastriate visual cortex and other areas of superior parietal lobe. And AIP, on the contrary, has strong connection, very strong connections with the inferior parietal cortex, inferotemporal cortex, but also ventral premotor cortex, prefrontal cortex. You see a different pattern of connection. So we think that AIP is more in the position to give the kind of input you are suggested, but is again a speculation derived from neuroanatomy in this case, and also the functional properties found for these areas.
Great talk. I wonder, you answered it in part to Carlo's question, but I wonder, given that MC and in the literature there are a number of studies showing that patients with V1 lesions are able to reach and grasp objects in their blind field, I wonder, have you explored the input from pulvinar to MT and potentially to 𝗍𝘁𝗍? I mean, what kind of contributions, extra nucleostriate contributions, could there be to these computations? And have you speculated about this or indeed investigated it? Yeah, we have found no connections with superior colliculus, but with the pulvinar, yes, and MT, not directly MT 𝗍𝘁𝗍, but very strong connections between V6 and MT. Very, very, very strong. And 𝗍𝘁𝗍 as a direct visual, or whatever, input on 𝗍𝘁𝗍. So probably the input from MT to 𝗍𝘁𝗍 could arrive indirectly through V6 or MST also, or other areas. And yes, I mentioned MC rather than the F or other patients because MC had really a massive lesion, not only restricted to LOC or very important places in the ventral stream. So you could guess that the full visual input is, geniculate striate input is missed then. And she could perform the task with other dorsal stream areas, and this one seems to be spared. This is. And just to follow up on that, MC, of course, shows size constancy for grasping, even though perceptually she has no size constancy or any kind of shape recognition. But I wonder, have you explored whether or not the neurons in 𝗍𝘁𝗍 are tuned to the real size of the object or the projected size on the retina? I mean, clearly for grasping, the most important thing is that you compute the real size so that you can conform your hand to the size and shape of the object, not to its projection. Yeah, you're right. We did not do. We wanted. We also spoke with Peter Jansen, who was doing in other regions, but we have not done. Yes, this would be very important to be checked.